little champions, I hope that you are doing awesome, you're wonderful, you're happy, you're healthy and everything is nice. Even if it is not nice, we are going to make it nice. So I welcome you all to PU2 Brahmastra series, but today we are going to deal with this chapter, that is biotechnology and its application, and time for a good story, that is thank you all for showing lots and lots of love to the previous video. I loved it. Okay, seeing you all commenting down there made me so happy. You can never imagine how happy I was. Okay, at least a few comments were there. So whenever you say, ma'am, your lectures are helping us, so I believe that basically apart from appreciating me, you are also appreciating yourself for the efforts you're putting in to watch a lecture. Basically you are studying, right? So that means a lot to me that you're putting down your efforts, my dear children. Let me tell you, at the end of this, okay, everything, there's only one thing that basically fuels up the world. Okay, there's only one thing that makes the world keep going, and that is love. So keep on showing your love to us. We also need a little amount of motivation. Keep doing that. Whatever you are doing, something positive you are doing, keep doing that. And also definitely know us your problems also in the comment section. We are there to help. Okay, I don't think I have left any comment unread or basically unreplied. I have tried my maximum to reply to all the comments. So definitely keep on doing that. Keep on asking me questions, keep on telling good things, keep on telling bad things. I will be replying for everything that you are saying. Yeah, and also share it with your videos, share it with your friends. Okay, I'm not talking just about this video, any video that you think might be helpful for your friend to study. Apart from this channel also, there are other areas also where you get videos. Wherever you see a video which you think could be helpful for others, do that. Help, love, do everything positive that you can do. You are going to ace, my dear children. So let's get into the topic. So over here, biotechnology and its application holds 6 marks, my dear children. According to blueprint 1, there will be one 1 marker and one 5 marker question. So you can expect a 5 marker question coming off from this particular chapter. And according to blueprint 2 and 3, it says that there will be one 1 marker, one 2 marker and one 3 marker. Okay, no 5 marker, chill. Now, if you are someone who is interested in getting into this particular series, that is Success Blueprint Test Series for PU2 children, definitely do not forget to enroll. The link is given in the description box over here. The test series have already started from 15th of December, but you still can enroll. You will still find the questions and everything. You'll still find the solutions for that. So please don't worry. And it's all just for $5.99. So do not miss this chance. And how many questions have I included over here? 10 1 marker, 2 2 marker, 3 3 marker and 2 5 marker. Okay, so this includes your model question paper questions and also the questions which were repeated the previous year. So let's get into it. Tell me the answer before I do. Okay, DNA in a clone of cells followed by detection using autoradiography is called. So you know you're talking about DNA and you are saying that basically you are able to detect this DNA using autoradiography. There's only one type of DNA which you can detect using autoradiography, and that labeled DNA is known as, my dear children, a probe. So probe is what? A radioactively labeled DNA, which is why it will be showing off a particular color under the autoradiography machine. Cool? So what do you call this? You call this as a probe. And why do you need a probe? Basically, I am not able to find the DNA over here. Okay, with my naked eyes, I cannot see the DNA. But if I let the probe go and bind and find its complementary pairs over there, I will be able to understand that, okay, here I have the DNA that I am looking forward for. You get it? So this becomes your answer. This is a model question paper question. Okay, next question. So this is the probe that I was talking about here. You see bands. These bands are visible because of autoradiography machine and also because they are labeled. Now, use of bioresources by multinational companies and other organizations without proper authorization and compensatory payment is referred as. So I am basically using some bioresources. Okay, probably some type of rice or something. Okay, I am a owner of a big multinational company. Okay, so I am actually borrowing it from some other nation and I am using it, and I am saying that, okay, I have the proper authorization, I have basically all the rights to sell it and things like that, but I am not giving any compensatory payment to the people I bought it from. This is known as what? This is known as biopiracy, biofortification, bioprospecting, bioprocessing. It is biopiracy, my dear children. So use of resources by multinational companies and other organizations without proper authorization and compensatory payment is referred to as biopiracy, and people who do it are really bad. So please not indulge in any such thing even if you grow up, okay? This is a model question paper question. Going ahead. Select the correct match. Gene is given over here, target insect is given over here. So gene cry1AC, cry2AB, cry1AB is given, and the target insect says this particular gene is for this particular insect. Do you think is it true? What do you mean by this particular gene first of all? So cry gene, no, cry gene is found in Bacillus thuringiensis. I might be wrong with the spelling, but please don't mind. It is also known as Bt, which is a bacteria. So this gene is found in a bacteria, my dear children. So what happens is that basically there are certain insects which goes and feeds on what? The plants. Suppose the bacteria guy is also here and the insect is coming and feeding this particular plant. But now accidentally it has taken in the bacteria as well. It has taken in the bacteria as well. Now what happens? Now what happens? Basically it has suppose reached to the gut of the bacteria and gut of the insect. Now here we have the bacteria. What happens is that the bacteria in its body have some prototoxins. Have some prototoxins, not protoxins. Wait. It has certain prototoxins, that means inactivated toxin. Just as it enters the gut of the insect, what happens, my dear children? These prototoxins become active. They become active because of what? The alkaline pH. The alkaline pH of the gut. Now when they are active, they are going to create a ruckus over here in the epithelial cells of the gut, and later this particular insect is going to die. Okay, so the gene which codes for this particular toxin is known as cry gene. So each bacteria that you see is going to release specific type of toxin for specific type of organism, insect. So you have cry1AC, which is going to actually target cotton bollworm. Cry1AC targets cotton bollworm. Cry2AB also targets cotton bollworm, but it is written corn borer, so this becomes wrong. Cry1AB targets corn borer, but it is written cotton bollworm, so this is also wrong. So how do you remember it, ma'am? I am not able to remember it. The answer for this is what? There is only A is correct. A is the only one that is correct. But how did you come to that conclusion, ma'am? I can tell you I don't know if it's a good way to remember, but I remember it like this. So cry, then 1, and then cry, and then 2. So A, C is there, then comes A, B. So the first one, the first letter will be having C, so I'll write cotton. The second letter will be having B, so it becomes cotton ball worm. Okay, now you say corn borer also is CB, ma'am. Then what will we do? So for that, I don't have any specific way, but it is like cry1AB. I remember like that. So C and B over here. What is this? Corn borer. This is how I remember. So cotton bollworms, and second part of this is this, which is why we say cotton bollworm B. Second part of this starts with B, so I wrote bollworm in the second part. Here basically it's C and B, corn borer. I don't know if it makes sense, but this is how I remember. So both of these guys are for cotton bollworm. This guy is just for corn borer. Okay, so this becomes your answer. Cool. Let's get into the next question. Which of the following statement is correct? Most Bt toxins are insect group specific. You just saw right now. They are kind of very specific to whatever insect is there. So this is correct. RNA interference takes place. RNA means RNA interference. Takes place in all eukaryotic as well as prokaryotic organism as a method of cellular defense. Let me tell you one very important thing. RNA interference basically, just simple terms I'll say, okay. Suppose there is one organism. There is one particular organism who is the bad guy. Who is the bad guy? Okay. This particular organism is basically going to have its own RNA, and this RNA helps in the formation of what? Protein. And this protein is kind of really bad for what? For the plant, my dear children, which is why I called it the bad guy. Now, RNA interference is that technique where basically what I am going to do is, this is a single stranded RNA which undergoes a process that is known as translation, to form a protein. It always has to be single stranded. What if I make it mingle? I do not state to be single, but I make it mingle. So what do I do in RNA interference is that basically I am bringing out certain changes in the plant such that the plant has the pakka complementary pair, complementary as in the pakka opposite pair of this RNA. Okay, so there is a double stranded RNA over here, children. There is what? A double stranded DS RNA over here. So when the bad guy is going to eat the plant, is going to feed on the plant, what is happening? In their body, you get this double stranded RNA. And already over here, what does it have? It has a single stranded RNA. Just as it sees this double stranded RNA, one strand of it comes and joins over here and makes the other strand which is there, it gets degenerated. The other strand gets degenerated, one strand comes and join over here. Now what will happen? The single stranded RNA which was there, is it now single anymore? No, it is mingle. So basically it has become what? Double stranded RNA, my dear children. Okay, this is known as RNA interference. Basically, you are not going to allow the RNA to make the protein with the help of translation. Translation always requires single stranded, but now it is double stranded. So you are interfering between this particular process. This is known as RNA interference. Now, if it is able to do that, then will it make the protein? No. If this protein is not being made, will the plant get affected? No. So this bad guy is actually now safe because it is jailed. It's all the things that it was doing has now been found out, and there is no way it can get out. So RNA interference takes place in all eukaryotic. What do you mean by eukaryote, children? We are eukaryotes we people who have a cell, and the cell has nucleus. The nucleus is bound by a membrane. Also we have other organelles which are bound by membranes, are known as eukaryotes. Whereas organisms which basically do not have a proper nucleus, their DNA is just lying around there, okay, in the cytoplasm. They do not have a membrane bound organelles and stuff like that. Those organisms are known as prokaryotes. For example, bacteria. So RNA interference is generally a cellular defense in the case of eukaryotes, not prokaryotes, but over here it is written eukaryotes as well as prokaryotic organism. So this becomes a wrong answer. Eukaryotes is the only correct thing. Okay, the next thing, the recombinant therapeutics do not induce unwanted immunological responses in humans. It does not, because it is kind of these recombinant things that which are there, it is kind of taken from like somewhat has reference to our body also. So our body does not consider it as a foreign thing, which is why our body would not be producing any sort of response, unwanted response. Now, sometimes it does, but mostly it does not. So this is also correct. Adult onset diabetes can be controlled by taking insulin at regular time intervals. Yes, so person who has diabetes, okay, in adult stage, diabetes is what? Basically when the level of glucose in our blood, it goes really high. So at that point of time there should be someone to lower down this level. Who is that person? Who is that thing? Not person, basically a hormone. What is that hormone? It is insulin. Insulin basically says, come here, all the glucose molecules, I am going to make it come together and form what? A glycogen. So basically the level of the glucose in our blood goes down when it's there. So this diabetes disease can be controlled by taking insulin at regular time intervals. That is also correct. So this exactly becomes the wrong statement which is being put over here. Cool. Let's go on to the next one. Which of the following statement is incorrect about the gene therapy in ADA deficiency? What do you mean by ADA? ADA stands for adenosine deaminase. Adenosine deaminase, which is an enzyme which is very much needed in our body. Otherwise, in our body there will be accumulation of something which is not needed very much. Okay, so this accumulation might cause later very very, very uncomfortable situations, okay, immunological problems in the human body. So basically you have to find out what is incorrect about gene therapy in ADA deficiency. Gene therapy, you know what it means. Basically I am going to make certain changes in the genes, I am going to put some extra DNA or something like that, and I am going to treat a particular disease. That is known as gene therapy. So what is incorrect about gene therapy in the case of ADA deficiency? So lymphocytes from patients' blood are taken out and cultured. What do you mean by lymphocytes? The one type of WBCs, they are one type of WBCs. So what happens is that in gene therapy, so the people who have adenosine deaminase deficiency, I'll be taking out certain lymphocytes, and then in these lymphocytes I am going to put the DNA. I am going to put the DNA because they are missing the DNA which produces what? Which produces adenosine deaminase. So I am going to put that DNA, and later basically what I am going to do is that I am going to put it inside the human body again. Put it inside the human body again. Okay, that is the thing that I am usually going to do. So that I have the gene over here, and it will produce its product. That is adenosine deaminase. So lymphocytes from the patient's blood are taken out and cultured. It is cultured. A functional ADA cDNA. What do you mean by cDNA? Complementary DNA. Okay. ADA cDNA is introduced into these lymphocytes. So I am going to basically put the gene for ADA into these lymphocytes. Lymphocytes are then reintroduced into the body of the patient. After that, the lymphocytes are being put down back into the body of the patient. Now, patient do not require periodic infusion of genetically engineered lymphocytes. It says that patient, suppose I put it in this particular patient. Now this patient does not require lymphocyte infusion, as in it does not require more amount of lymphocytes every now and then. Is that so, children? Is that so? Do you think that this is a perfect cure that we are having over here? Do you think that we will treat forever? No. So that is a sad reality. Okay, that is a sad reality. Basically, the people who are having this deficiency have to take periodic lymphocyte infusion. So every time lymphocytes are taken in, again gene therapy is being done, and then it is being put in the human body, and the same cycle repeats again and again and again. So this particular thing is entirely wrong. Okay, patient do not require infusion of genetically engineered lymphocytes. That is wrong. They definitely require it, because these lymphocytes that you see over here, they also die. They also have a lifespan in our body. They are not immortal, my dear children. They are mortal. They will be dying. So obviously after they are dying, who will be giving giving codes for this particular protein, or basically for the enzyme adenosine deaminase? So that is the thing. Cool. Next question. Match column 1 with column 2 and select the correct option from the given codes. Okay. Alpha-1 antitrypsin, what is it, children? It is a very important protein which is being produced with the help of transgenic animals. That means in the animals we are going to put the gene for this particular protein, and the animal is going to produce it, and then we are going to later take that product and use it for our own thing. So why do we use it for? Alpha-1 antitrypsin is basically used to treat this particular disease which is known as what? Emphysema. It is known as what? Emphysema. Now, let's look for something that is more easy, that we all know. ELISA, we have studied in human health and diseases. ELISA is enzyme-linked immunosorbent assay. Why do we need it for? For finding which disease, children? AIDS, HIV AIDS is found with the help of ELISA. Now, retroviral vector. Why do you think we need retroviral vector? Again, we studied in biotechnology principles and processes also. So we have different type of vectors. Vectors are those organisms that will be able to carry the DNA into the host. Okay. So for example, I needed, okay, in the lymphocytes which were there, I needed the DNA which was coding for adenosine deaminase. I cannot just randomly basically inject this particular DNA. Instead of that, I am going to use a retrovirus. A retrovirus. This just disarmed. That means it is not going to cause any trouble. I am just going to use its machinery. So what is the machinery of the virus? Viruses, what do is that if they have some genetic material, they will come and basically insert the genetic material into our genetic material. So we are going to use their machinery to put my gene of interest over here. You get it? And this treatment method is known as what? Gene therapy. We just studied about it, right? It is known as gene therapy. So retroviral vector, always remember retroviral, this is lymphocytes. Lymphocytes are human cells, my dear children. They are human cells, and whenever human cells are being considered, just understand retroviruses are being used as vectors. So retroviral vector is used for gene therapy. What are you left with? We are left with transposons. Transposons are those segments of the DNA which basically what do they do is that they move from one area to the other area. They are known as jumping genes. They are known as jumping genes. So transposons are basically what? Mobile means movable, right? Mobile genetic element. So they are the ones that move from one area to the other ones. So transposons are mobile genetic element. So alpha 1 antitrypsin is for 3. A is for 3. So we have two options saying that A is for 3. Let's delete the other options. Now transposon is 4th, mobile genetic element. Transposon is 4th. So we have one option with that. Let's see the other answers. C is for 1. Okay, C is for 1. And D, retroviral vector, is for what? Gene therapy. So this becomes the answer, my dearest little cuties. Works? Chalo. Next, I hope you're understanding. Are you understanding everything, children? Do you have doubt? Do you have doubt? Please feel very free to ask me any sort of doubts that comes through the class, okay? Now, read the given paragraph and identify X and Y. Okay, X is the right granted by a government to an inventor to prevent others from commercial use of his invention. What? X is what? A right that is granted by a government to an inventor. For example, you are the inventor. It is a right granted by you, to prevent what? Others. To prevent others from commercial use of your invention. So you invented something, and now the government is giving you some right that none of the other people can use it. To prevent them others, okay. Now X is Y. X are granted for biological entities and for products derived from them. These are called Y. When X are granted for biological entities and for products derived from them, then these are called Y. Okay, when whatever right you have got, okay, that particular right is for the biological entities, for the products that you are deriving from them, then what do you call this? Basically, X is what? The right given to you, it is known as patency. You have the patent that no one else can use it. And now when this particular patency is for biological contents, what do you call this as? Biopatent, my dear children. So this becomes the answer. Now just give me a second, I'm coming in a second, okay? Okay, now moving on to the next question that we have over here, which is, the phenomenon of silencing of a specific mRNA due to complementary dsRNA, that is double-stranded RNA molecule that binds to mRNA and prevents translation is. What is this phenomenon known as? We studied it. Remember, we studied it. There was an mRNA which was single and we made it mingle, right? What was it? What was it, children? What do you call this particular process as? You call this process as RNA interference. Okay, what do you call this as? You call this as RNA interference, or you also say it like this. Okay, where you're going to prevent the translation. We are going to learn about it once more. Okay, there's a question regarding it where I have explained each and everything. We will come to that as well. Cool? Now, the technique that serve the purpose of early diagnosis. So most of the time we people are not able to do the early diagnosis of lots and lots of diseases, but there's a technique which could help us do that. And why is the reason? Why are we not able to have a clear early diagnosis as well? First thing, we do not have the symptom. Second thing, the level of that particular thing in our body might be really less. So there is no much techniques that could help us identify that little amount of the thing which is there in our body. So there is a process which could actually help you with that. It recognizes even a little amount of DNA, whatever genetic material is there, and later amplifies it. And makes it really easy to understand which disease is there in that particular animal, organism, or basically human being. So the technique that is used, which works on the principle of amplification of the genetic material, is none other than PCR, or polymerase chain reaction. PCR, or what do you call it? Polymerase chain reaction, my little young people. Cool, always remember, early diagnosis, PCR. Now going ahead, mention a gene that codes for insecticidal protein in Bt cotton. Insecticidal as in a gene that is going to code for insect killing protein in Bt cotton. So in Bt cotton, that is Bacillus thuringiensis. Okay, Bt cotton means that particular gene of Bacillus thuringiensis is put in cotton. That is what you mean. Okay, so what is that particular gene, my dear children, which was coding for the protein? It was known as cry gene, because it is going to make the insect cry really bad. So this becomes the answer, and with this we are completing all the 1 marker questions from this particular chapter, all the important 1 marker questions from this particular chapter. But feel very free to solve more questions as well. But I have included all the questions which have been put in the model question paper, and also certain very repeated questions that have come. Okay. And rest of the things we would be studying in the 2 marker, 3 marker section. So whatever you study in there, you can frame your own MCQ questions out of the 2 marker, 3 marker as well. Works. Chalo. First question of 2 marker category is, ELISA is one of the methods of molecular diagnosis. What is the principle of this technique? Name the disease which can be detected by this method. So we know the disease, that is HIV/AIDS. Now what is the principle? How does it actually work? So it actually works on the basis of something that is known as antigen antibody interaction, my dear children. Antigen antibody interaction. So I hope you know that these guys are friends. Okay, these guys are friends. Okay, for example, if antigen is of one type, for example type 1 antigen is there, then it will bind with only type 1 antibody is there, then it will bind with only type 1 antigen. Okay, so they are made for each other always. They are made for each other always. They do not cheat, they do not have any other partners or anything like that. Okay, so basically what happens is that, suppose I have a well like this, and I have certain antibodies which I have taken from. I already know that this antibodies are being produced in a patient who has HIV/AIDS. So I already know about what? That these antibodies which are there are found in patients who have HIV/AIDS, because our body produces antibodies to fight against it, right? So obviously I have taken it out and it's there with me. So I am very sure about these antibodies are for HIV/AIDS patient. Now suppose I have a patient and he or she is unsure that whether he or she has AIDS or not. So I am going to take their blood and I am going to take their antigen, or basically I am going to make the antigen react with the antibodies. Suppose it was not, for example, I call this type 1 antibody. Now suppose it was not the one that could bind with type 1 antibody, it won't bind. But suppose this particular person's antigen comes and binds over here like this. That means what? This particular antigen is there in the body. That means here later on probably this body will also be producing antibodies like this. Okay, that means this particular person is infected with HIV/AIDS. Okay, because the antigen have come bound with this antibodies. If it was not of the same type, it might have not have bound, but it is binding. But still we are not able to understand whether it is binding or not. So we basically have the next set of antibodies that we have only created. Okay, and these antibodies, no, they are linked to some enzyme. They are linked to certain enzymes. So these antibodies again will be only coming and binding if the antigen is placed over here. Will be coming and binding to the antigens only if it is there over here. And these all are linked to what, children? It is linked to enzyme. It is linked to enzyme. Now you might be thinking, what's the use of it, ma'am? Basically, now I am going to treat this particular thing with some substrate. Now, the enzyme seeing the substrate will be bringing out a color change over here. That means here the antigen have come and bound, and in response to that the secondary antibodies which were there, they also came and bound, which is why the enzymes which were associated with them, they became active and basically acted on the substrate to give out the color that is being shown up here. That means this particular person has HIV/AIDS. Okay, there are different type of ELISA. This is known as sandwich ELISA, because basically you have 2 type of antibodies and in between you have antigen. Okay, so enzyme linked immunosorbent assay. ELISA stands for enzyme linked immunosorbent assay. It works on the principle of antigen antibody interaction. A specific antigen or antibody binds to its corresponding counterpart. This interaction is detected using an enzyme-linked antibody. How do you detect it? Using an enzyme-linked antibody, an antibody that is linked to an enzyme. And what? And a substrate which produces a color change indicating the presence and amount of target molecule. So the color change is way too much. That means whatever target molecule is there, it's way too much as well. You get it? That's how we are able to work with the help of ELISA. This is used to detect HIV/AIDS, my dear children. Chalo. Next question. Why are meristems preferred in tissue culture? What do you mean by tissue culture, children? Tissue culture basically means, for example, I have a plant, okay? I have a plant. I take the tip of the plant over here. This tip basically are made up of certain cells which are known as meristematic cells, which has the ability to keep on dividing. They have the ability to keep on dividing, my children. So I took this, okay, I took this and I call this as now as an explant. I took this. This is known as explant. Now, usually nourishment and everything was provided with the help of the plant body itself. But now since I have separated it, I have to put this explant in one particular beaker with nutrient medium. With what? Nutrient medium. Using this nutrient medium, this will keep on dividing and make a larger bunch of tissue, which will now be known as what? Callus. Which will now be known as what? Callus. Explant was the first name, now we call it as a callus. Now we put the callus in a medium that contains the rooting hormone, a hormone that will help in formation of root. Slowly this callus will be forming roots. Then I will be putting it in a hormone, in a medium that has shooting hormones, which will basically help this callus to form shoot as well. Then once root and shoot is being formed, I am going to put this particular plant into what? Into soil, and now I have a new plant over here. So why do you think this meristem is being used in this particular process of tissue culture? Why do I say tissue culture? Because I am taking a group of cells, and group of cells are known as what? Tissue, and culturing it, right? So why meristem is being preferred? First thing I already told here itself. It has the ability to divide very rapidly. It has quick division ability. Apart from that, children, meristems are generally virus free. They are generally virus free. So I do not expect my plant to be affected by any sort of diseases as well. Cool? So that's how it works. This is how you have to write. The meristem is free from virus, and meristematic cells have the capacity of cell division. If you want, you can make the whole thing as well. Okay, but that is not necessary. It's a 2-marker question. So just stick to the answer, write it in points. Okay? Meristem, right, they are found at the apex of the root and the shoot. Always right. Meristems are found in the apex. Apex means tip of the shoot and the root. And then say that they are virus free, and meristematic cells basically have what? The capacity of cell division. Cool? Now, okay. This is a 3-marker question, my dear children, that we have over here. How did an American company named Eli Lilly use the knowledge of rDNA technology to produce human insulin? What do you mean by rDNA? Recombinant DNA. And I am combining 2 different types of DNA. That is known as recombinant DNA technology to produce human insulin. So are you aware of this thing? Eli Lilly is still there. My friends keep on trying for the job, for getting into jobs, in Eli Lilly company, which is a very reputed company. Okay. So what did they do? How did they use their knowledge of rDNA technology, recombinant DNA technology, to produce human insulin? So human insulin, as I said, what it does, it takes in all the glucose, says you come here, you come here, you come here. I am going to tie you up and make glycogen over here. Because if you guys are free, basically you are going to increase the blood sugar level. I do not want to do that. So insulin is for that, but some people don't. Some people may not be able to produce little amount of insulin that they should be producing. The pancreas might have some problem. The pancreas is a gland which releases insulin. So in that case, we have to inject insulin from outside source. So usually earlier we used to take pig insulin and stuff like that, but then later it was known that some people were kind of very allergic to all this thing. So then this company came up and said that, okay, we can try to make the insulin. But the problem with that was, there was one big problem, and that was, if you see a insulin chain, this is a pro-insulin chain. Okay, why do I call it pro? Because it is not active insulin. So over here they have 3 chains, my dear children. They have 3 chains. 3 polypeptides are there. Polypeptide means group of amino acids coming together. Okay, so I say 3 different polypeptides are there, 3 different types of polypeptides are there. One is known as A, one is known as B, and this long black one is known as C. Okay, whenever C is there, that means this is not active form of insulin. In our body, it is being produced like this, proinsulin. But then later, with the help of the enzymes which are there in our body, this cut, there is a cut being made over here, there is a cut being made over here, such that A and B are free now. And they can make disulfide bonds and things between them. And now, this thing that you see over here, it is known as what? Insulin. But the problem was that, okay, if we want other organisms to produce our DNA, we have to make sure that they are going to cut it like this also. They are going to cut it like this. So initially it was thought that, okay, we can take the help of one particular bacteria, that is E. coli, Escherichia coli. Okay, Escherichia coli, it's a bacteria, so it has plasmid in it. So they have their own chromosomal DNA. Apart from that, they have extra DNA also, which do not have any function. So if I put my gene which codes for A, B and C polypeptide, it will be producing that. But the problem is that if I produce the gene which is directly for proinsulin, if I put the gene which is going to produce proinsulin together, the problem is that the C part of polypeptide would not be cut. It would not be cut because this E. coli that you see, it's a bacteria, and bacteria, my dear children, comes under the category of prokaryotes. So they are kind of really less advanced than us. So they cannot cut the C DNA which will be also formed. Okay, over here. So what they thought was, LLE thought that, why not? We separate the genes also. There might be one gene coding for A, there might be another gene coding for B, right? So why not we put one gene over here in one Escherichia coli, and this will be coding for the A polypeptide. There will be another Escherichia coli, in their plasmid I am going to put the B gene. I am going to put the P gene, and hence the B polypeptide will be produced. So A polypeptide is produced, B polypeptide is produced. Right? Now the only thing that I need to do is join them. So after the polypeptides are being produced, it is being joined with the help of the laboratory thingy. Okay, it was done in the lab. The joining part was done in the lab. Okay, so now they didn't have to actually wait for the Escherichia coli to develop a mechanism to cut the seed part and things like that. They thought, why not? Find the gene, put it over here. Find the gene for B, put it over here, and make it separately. Let us join it later on. And that's how the human insulin, or basically humulin, was made. Human insulin or humulin was made. So the one that has C polypeptide is known as proinsulin, and the one that does not have it is known as just insulin. This is how you have to write. Isolation of insulin gene. The gene for human insulin was isolated from human cells. First thing, we have to take that gene out, right? Insulin is made up of 2 polypeptide chains, A chain and B chain. Insertion into vectors. Using recombinant DNA technology, the genes for A chain and B chain of insulin were separately inserted into plasmid vectors. What are plasmid? This thing that I was saying. Inserted into plasmid vectors. I am putting it here, okay? First of all, I will be taking out the plasmid, and then I am going to put it, and then I am going to later put it back again. So first thing, I am going to take it out, the plasmid. I am going to insert my DNA of interest, okay, and then I am going to put it back over here. Okay, that's what I have to do. Into plasmid vectors. Now, transformation into bacteria. The recombinant plasmids carrying insulin genes, the recombinant plasmid, the new plasmid which is being formed, the newly combined plasmid which is there, okay, carrying the insulin genes, it were introduced into E. coli. They were introduced back into E. coli, that is the bacterial cells. Now what? Production of insulin chains. The bacteria were cultured in large bioreactors. They were put in large bioreactors, my dear children, okay, and they were cultured such that they produce A chain and B chain of human insulin separately. Later on, what has been done? Purification and assembly. The 2 polypeptide chains were extracted, purified and combined using disulfide bonds. Okay, disulfide bonds were being made, and then that's how we got the active human insulin. So this is how the Eli Lilly company worked. Cool. So this is a very cool experiment being done by them, and it has they have gained lots and lots of love for that. Sorry. Next question. One of the applications of biotechnology is to get pest-resistant plants. Justify the statement with reference to Bt cotton. So one of the application of biotechnology, the use of biotechnology is to what? Get pest resistant plants. What do you mean by pest resistant? Basically, if I am a plant, I am resistant to pest. As in, any insect will come and eat me, basically there won't be any effect in me. That insect might die, but not me. So that is pest resistant. See, okay, I am resistant to that particular pest. Okay, so justify this in reference to Bt cotton. So Bt cotton, if you know, is a pest resistant plant. Is a pest resistant plant. So how do you think it has become pest resistant? Basically, the gene that is cry gene, which was there in the bacteria, has been taken from this particular bacteria and has been inserted inside the plant cells. Inside the plant cells. Now what will happen? In the plant cell also this gene is there. It is going to produce the same toxin, the same prototoxin which is there. Now if an insect comes and basically tries to feed on this particular insect is going to die. That is why it is now known as what? Bt cotton. How do you write the answer? You can write it like this. Some strains of Bacillus thuringiensis produce protein crystals, insecticidal protein that kills insects, from lepidopterans. Lepidopterans are the ones where you include butterfly, moth and stuff like that. Okay, coleopterans are there, which includes I forgot, beetle, yes, and then dipterans are the one where you have flies and all. Okay, so there are different categories. So some strains of Bacillus thuringiensis, as in some type, okay, there are different type of a bacteria, that is Bacillus thuringiensis. Some strains of them, basically they are going to produce protein crystals, which are, what are these protein crystals? They are insecticidal protein, that is insect killing proteins, okay, that kill insects from this category. Now protein crystals form during certain phase of their growth, okay, they are saying it is being formed during certain phase of the growth. The cry gene, cry 1AC and cry 2AB from B thuringiensis coding for the toxins are transferred to cotton plants. This gene is being put in the cotton plants. Proteins encoded by this gene control the cotton bollworms, I told you. So here they specifically spoke about what type of insect is also being controlled. So cry 1AC and cry 2AB is going to control cotton bollworm. If you remember, I told you the trick. So cry 1AC, cry 1AC. So first letter is starting from C. Now cry 2AB. Second letter is going to start from B, that is cotton bollworm. Okay. Now Bt cotton plants express the Bt toxin gene by producing toxin that provides insect resistant. Bt toxin protein exists as an inactive prototoxin. Insect digestion. Then you have to tell the story of what? The insect who is going to digest it. So insect basically digests this. And what happens? The inactive toxin becomes active. Why? Because it is inside the gut of the alkaline pH. It is inside the gut where there is alkaline pH. And this alkaline pH makes the inactive things really active. And the toxin then binds to the surface of the mid-gut epithelial cells. Epithelial cells are the one that lines the gut. Okay. And then create pores that causes cell swelling and lysis. Cell swelling as in the cell will swell and then lyse as in break open. Okay, which will obviously lead to the death of the insect. So you have to say about Bt first, Bacillus thuringiensis, that they are going to produce some toxin, and this toxin is coded by some gene which is known as cry gene. You can give example that cry this and this is coding for a gene which is basically going to kill cotton bollworms. Then you can say the action plan, how does it exactly work, and that's how you frame your answer. Cool? Chalo. Next question. How is ADA deficiency cured by gene therapy? ADA, adenosine deaminase, that's an enzyme which is really important for our good immune response. Okay, for a good immune response. How it is cured by gene therapy, I already told you. Basically, here when someone is saying that they have ADA deficiency, that means their DNA do not have that particular gene which is going to code for adenosine deaminase. They do not have this particular thing. Might be some alteration or altogether it might not be there. So what do you do in this case? You do gene therapy where from this particular person, the lymphocytes are being taken out. Lymphocytes are type of WBC, white blood cells. Okay. Now lymphocytes, basically they are infected by retroviruses which are going to act like vectors. So I have made sure that this particular retrovirus has my gene of interest over here. So when it is going to affect these particular cells, basically what happens? Suppose this is the genetic material that was there already in the lymphocyte. Now when the retrovirus comes in effect, basically it is going to put its own genetic material also. That means it is going to transfer the thing that I was actually needing. And now the lymphocytes have this thing. All the lymphocytes which are basically there, they have what? This particular DNA that is going to code for what, children? Is going to code for adenosine deaminase, which is an enzyme. And enzymes are proteins. So genes are coding proteins. So this gene is also going to code for a protein. Now after this is being done, okay, after this transformation has been taken place, what I am going to do? I am going to take this lymphocytes and then put it back into the human body. But I have to make sure that this particular person has to undergo what? Regular transfusions. This particular person has to undergo regular transfusions. Cool? So that is there. Because lymphocytes, they die after some time. They have their death after some time. So please remember that. Okay. Now one very important thing, okay, the DNA which I am going to put over here, you always call it cDNA. You might have seen it. You might be having this confusion, what is cDNA? cDNA stands for complementary DNA. Complementary DNA. Listen to me very carefully, okay? Whenever I say that a DNA is responsible for a gene is responsible for protein, what happens is that the DNA is first transcribed to something that is known as mRNA, messenger RNA, which is single-stranded, okay? Which is what? Single-stranded. And this DNA strand which is there, no, DNA has lot of areas which code for something, which do not code for something. So whatever coding thing is there, okay, that comes together and makes the mRNA. So that does not have anything that does not make any sense. It does not have any nonsense thing in it. What is it? mRNA. Normal DNA might have lot of region that does not code and things like that. But mRNA has only the important stuff. And thus mRNA is now going to make protein. So what is cDNA made out of this? How is cDNA made out of this? Basically this mRNA, usually DNA makes RNA, but this time RNA is used to develop a complementary DNA over here, which is known as cDNA, such that basically inside my body I don't have to cut it and do anything. I have the ready-made content. Over here I don't have the ready-made content. So I am taking cDNA. How? I am going to reverse transcribe. This process is known as transcription. This process that is there is known as transcription, where basically from DNA I am making mRNA. But the opposite of it, if it is going ulta, what do you say? Reverse transcription is there, where I am going to make a DNA out of it. I am going to make a proper DNA that has particular codes, okay? Particular codes. You might be thinking, how did we get this mRNA? It is not a direct process. There are so many other things also. Then hRNA is first being made, then it is being cut, spliced and everything, and then you get mRNA. So don't think that it's just making the copy of this thing only. It's not like that. There are so many things. At the end you get mRNA, and now I'm going to reverse transcribe it and make a complementary DNA out of it, and that DNA I'm going to put it over here, such that there is no waste thingy over here. Okay, that is the use of cDNA. I hope now you understand. So so there is a background thingy that is given in the answer. I want you to just have a read. Okay, I'm not telling you to put it in your answer, but please have a read. The first clinical gene therapy was given in 1990 to a 4-year-old child with a girl child with adenosine deaminase deficiency. This enzyme is crucial for immune system to function. The disorder is caused due to the deletion of the gene for adenosine deaminase. There is a deletion of the gene. That means that the particular gene is not there itself. Okay, how it is caused, children? Please remember, deletion of the gene. So these small informations are really important when it comes to MCQs, which is why I made you read it. Okay, now in some children, ADA deficiency can be cured by bone marrow transplantation. Okay, so bone marrow is a place where all our cells and everything is being made. So in some people it can be cured basically by having a bone marrow transplantation, so that in bone marrow whatever stem cells are there, okay, if it has a DNA, it will give DNA to all the cells which is made by them. So that bone marrow transplantation can be done. In others, it can in others it can be treated by enzyme replacement therapy, where what is done, basically a functional ADA is given as an injection, that is known as enzyme replacement therapy. But the problem to both of these approaches are they are not completely curative. Okay, this does not completely cure a person's what's the situation. So there comes the role of this particular thing. There comes the role of this particular thing, but again this is also not completely curative. The things that we were talking about, the lymphocyte and everything, it is also not completely curative. There is only one thing that is completely curative, that is, for example, when during embryonic stage, okay, during embryonic stage itself, if I am getting to know that this particular has a disease, and if I am going to put the cDNA in the embryonic cells, then what happens in the embryonic cells, basically these cells are small in number, they are going to keep on dividing. So if I am going to insert in one particular cell, other cells will also keep on getting it. So in the embryonic stage only, if I do the gene therapy, it becomes easy. So that is, however, if the gene isolate from marrow cells producing ADA is introduced into the cells at early embryonic stages, it could be a permanent cure. So even this is not a permanent cure, but still works. Understood? All clear? Give me a thumbs up if you're getting to understand everything. Give me a thumbs up, my dear children. Now, going ahead with the first 5-marker question, which is very much interesting. Explain 5 benefits of creating transgenic animals. What do you mean by transgenic animals? Transgenic animals are the ones that have the DNA which does not belong to it. It is a foreign DNA that is being put into it. So the first and the foremost, okay, benefit of it is basically I am going to make so many products from it. For example, as I said, there is E. coli. If I am putting the gene for A chain or basically B chain, I am going to get my human insulin from that particular organism. So that is one thing. Similarly, there are so many proteins being made out of it. For example, alpha-1 antitrypsin, I told you, right? In the match the column question, it was there. It is used to treat emphysema. Emphysema is a respiratory disorder. Okay, it's a disease, not disorder, basically it's a disease. So basically, alpha-1 antitrypsin can be made in an organism and that can be taken out and that can be used for us. Okay, then we had the first cow, the transgenic cow, that was Rosie. Rosie was producing milk that had the same content like human milk. Human milk has alpha-lactalbumin. Okay, alpha-lactalbumin protein. So that Rosie also was basically, that cow, little Rosie, was also producing milk which had alpha-lactalbumin. So basically I put the gene over there, so hence it was producing it. So there are so many cases like that. Second thing, okay, apart from producing things for my own good, second thing, for curing diseases, for basically what? Curing diseases, I can create drugs and things like that from them, and also I can, which is not very moral of me to do, but yes, I can do drug testing also. As I said, it's not very moral of me, but that's how it works, okay? Please don't blame me, children. Please don't blame me, that's not me. Okay, so let's read it and let's understand it. I don't want to make it sound very bad. So that's why. So normal physiology and development. First, to understand normal physiology and development, transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of body and its development. For example, in the transgenic organism, in a bacteria or something, I can silence a particular gene and see what effect does it have on this particular organism. I'll get to know the function of that particular gene, and then probably if I get to know the function of that particular gene, it will be really important for me to understand some diseases or something else that is caused by it. You get it? So normal physiology and development is understood with the help of transgenic animals. Second thing, study of disease. Many transgenic animals are designed to increase our understanding of how we contribute to the development of disease. These are specifically made to serve as models for human diseases so that investigation of new treatments for diseases is made possible. So they are made like that such that we can treat and see that whether the disease is being cured and stuff like that. So cancer and stuff like that is something that is being studied like this for so many years now. Now, biological products, as I said, it can be made. Transgenic animals that produce useful biological products can be created by the introduction of the portion of DNA which codes for a particular product, such as human protein used to treat emphysema, that is alpha-1 antitrypsin, phenylketonuria, it's a disease, PKU, okay, and cystic fibrosis, that is also disease, it can also be treated. Now, human protein enriched milk, alpha-lactalbumin, is produced in Rosie, the first transgenic cow. Vaccine safety can be tested. Okay, so transgenic mice are being developed for the use in testing for safety of vaccines before they are used on humans, because we and basically the mice share a lot of common DNA. You won't believe, but yes, we do, definitely do. So transgenic mice are being used to test the safety of polio vaccine and stuff like that, and later on human beings, basically on human beings it is being tried. Now I know this trial and things like sounds to be very cruel. Actually it is cruel. Okay, it's not just sound cruel, it is cruel, but then there are so many things that are coming up which are making sure that there is no ethical problems that arises. Okay, so initially when I was in school and all, no, my brother was in school at that point of time, he was allowed to cut frog and this and that. When I came, okay, that thing was totally banned. That thing was totally banned. No one was allowed to cut frog and all, and I said thank you. And I said thank you, but apparently we had to do in MSc. But okay, this thing, slowly things are being improving, but also we have to find a new way of testing. Obviously we cannot test it directly on humans because, you know, okay, similarly we cannot directly test on them also. I understand if there is some animal lover, even I am an animal lover, I have a pet dog, so I understand it's not like that, but this is how it is. Okay, and things are being working, okay, things are being done to to a greater extent to avoid the use of these animals or basically organisms. So that is why bacteria and things like that are being used, but then there are organisms which has quite similarity to us, okay, rather than bacteria which is a prokaryote. So that's the thing. Now chemical safety testing. So this is known as toxicity safety testing. The procedure is same as that used for testing toxicity of drugs. Transgenic animals, okay, first what are they done, I'll tell you. Okay, so in transgenic animals they are made to be kind of sensitive. They are kind of made to be sensitive such that, okay, we get to know that basically even a sensitive person, if they are getting this particular drug, what kind of response will that they be showing. So basically according to it, the level of basically or the toxicity of the chemical substances are being controlled. Okay, so chemical testing is for that. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. Toxicity testing in such animals will allow us to obtain results in a very lesser time as well. So these are the few benefits. Okay, 5 benefits there are. So 5 benefits of creating what? Transgenic animals is a very important question, my dear children, have been repeated so many times. So please do not think of skipping this. Okay. Now, the very last question, as I said, RNA interference is there. I will be teaching you, so I keep my promise over here. So, what is RNA interference? How does RNA interference help to develop resistance in tobacco plant for nematode infection? So, how does RNA interference help in what? Tobacco plant. So, there is a plant, my dear children, and what is this plant? This is tobacco plant. And tobacco plant is kind of crying because there is one particular nematode. Nematode means there is a worm. Okay, there is a worm whose name is Meloidigan incognitia, which affects the roots of the tobacco plant, my dear children. Okay, and it is going to cause ruckus. So that's why it is crying. So how it is affecting? Basically, I told you Meloidigan incognitia is going to have a gene. This gene basically is going to code for an mRNA which is single stranded, my dear children. And then this mRNA is going to make a protein. And this protein is the one that troubles the tobacco plant's root. Okay, this mRNA is single stranded, this gene is being there. So we have to treat this disease, right? Obviously, otherwise the tobacco plant will die. So we thought of doing RNA interference technique, okay, where we are going to make the single mRNA mingle with another RNA. But that's not an easy procedure. Okay, we have to, anyhow, our ultimate aim is to have double-stranded RNA in the plant itself. Such that when Meloidigan comes and eats the plant, basically it gets the double-stranded RNA. But where will the plant get the double-stranded RNA from? So what is being done is that this gene that you see is there. This gene which is coding for this particular protein, this gene is being taken up and is being put inside a vector. It is being put inside a vector whose name is Agrobacterium tumefaciens. Agrobacterium tumefaciens. Okay, so Agrobacterium tumefaciens is a bacteria. It will have its own chromosomal DNA, okay, and it will have its plasmid DNA. This Agrobacterium tumefaciens, this is actually more khatra, more dangerous for the plant, because it has a type of plasmid which is known as tumor inducing plasmid, and a type of DNA which is known as tDNA. It is known as tDNA, or you call it transfer DNA. What this particular bacteria do is that it goes and affects this particular plant. It usually goes and affects this particular plant, where it transfers the tDNA into the plant cell. It transfers the tDNA into the plant cell. So I am going to make a quick change over here. Instead of using this color, Agrobacterium tumefaciens, I will be coloring it in red. Cool. Now the plant cell which is there, I am going to color it in green. So now what is happening? Usually this particular DNA is put into the genome of the plant, and that's how it works. But what if I take the gene which I got from the Meloidigoin incognitia and I put it over here. I put it over here instead of the tDNA. So tDNA is not going to affect, and anyways that particular segment of DNA is going to be inserted over here also. So this particular thing that I have got, this particular gene that I have got, which I am going to mark in white, which is something that I am going to put it over here. This, I put it over here, this guy comes and affects the plant cell and transfers the genetic material which is now over here. Now the plant cell will be making the DNA, will be having the DNA. With the help of DNA, what is it going to make, children? It is going to make RNA, and probably later on a double-stranded RNA. It is going to make a double-stranded RNA. Okay, it is going to make an RNA and also single-stranded RNA and also double-stranded RNA. Now this double-stranded RNA, when it has been ingested by the Meloidigoin incognitia, what happens to the children? Over here, there is a single mRNA and there is a double-stranded RNA. So here they break up, here these guys they break up, okay, they break up, and what happens, the one strand which is over here, the red strand that you see over here, it slowly comes and binds with the mRNA. It slowly comes and binds with the mRNA. So pre-existing one, suppose it is green in color, now it is forming a double-stranded RNA. Will it undergo translation to make proteins? No, my dear children, it won't be undergoing translation. Okay, and this other DNA which is there, it gets degenerated. Sorry, RNA which is there, it gets degenerated. I'll tell once more, listen to me carefully. So there is a plant, tobacco plant. It is affected by Meloidigoin incognitia. It affects the root. This is a nematode. Okay, this is a worm. So what it usually does is that it has a gene which codes for mRNA, and the mRNA codes for protein, and that protein affects the tobacco plant. But what if I take the gene and I'm going to put it inside the plant? I cannot put it directly. I cannot put it directly, which is why I need a vector. Who is the vector? Agrobacterium tumefaciens is the vector, where the tDNA, the infected part, is removed, and I am putting my gene of interest over here. I am putting my gene of interest over here. This gene of interest basically is getting transferred because this is a bacteria that basically is going to infect. So this is going to do the normal infection, but it does not know that it does not have the tDNA. Instead, it has the other gene. It goes and infects, gives the DNA. This DNA forms a double-stranded RNA in the plant cell, and now the Meloidigoin incognitia, when it has the double-stranded RNA, when it ingests it, what will happen, children? One strand of it goes and makes a pair with the mRNA, and the other strand degenerates. Once it is a double-stranded RNA that is being formed over here, it will now not be able to undergo a process that is known as translation. It would not be able to undergo a process that is known as translation. Works? Understood? So this is how things work. Okay, so that is about the RNA interference. How you can write it? You can write it like this. RNA interference means silencing of mRNA. It involves silencing of specific mRNA due to complementary dsRNA, that is double-stranded RNA molecule, that binds and prevents translation of mRNA. Steps of RNA interference. First thing, identify the nematode gene. Scientists first identify the genes in the nematode that are essential for its survival or ability to infect the plant, this particular gene. Now, using Agrobacterium tumefaciens to insert the gene. To deliver the silencing gene into tobacco plants, scientists use Agrobacterium tumefaciens. a bacterium that naturally transfers DNA to the plant cells. You saw that, okay? The identified nematode gene is inserted into the genome of the vector. That gene has been put over here. Second thing, this gene, the DNA sequence is then inserted into the plant's genome using Agrobacterium tumefaciens during the infection. Later, what happens? This is being inserted into the plant genome. Now what? Production of dsRNA in the plant. Once the DNA sequence is inserted, the plant begins to produce double-stranded RNA, dsRNA corresponding to nematode's target gene. Okay. Now, when nematode feed on the plant roots, they ingest a double-stranded RNA produced by the tobacco plant. This double-stranded RNA triggers RNA interference in the nematode, silencing the target gene in the nematode cell. As a result, what happens, children? The nematode can no longer function properly and cannot cause harm to the plant, and tada, everything is done. So how do you write the answer? This is a 5-mark question. Please make diagrams and help. It will help you understand as well. Or probably write in normal format. Just think of this particular diagram. Give a situation to all. Okay, so this is actually a villain, but this is not, this is a foolish villain. This is a foolish villain. I'm going to utilize this particular villain. Okay, and this is a clever hero or heroine, whoever you want to have in your story. That. Okay, cool, works. These both are actually villains. So these were the questions from this particular chapter, my dear children. I hope the questions are done, and now we have come to this particular slide. But before that, if you think that this particular video was useful for you, what do you have to do? You have to comment down that, ma'am, it was a good class, okay, we understood things. And if you think that this class was not up to the mark, okay, you didn't understand certain things, say, ma'am, not good. It's not fine. You have to improve. It's okay. I'll still improve. And also one more very important thing, if you have any doubt, please put it down. And your exams are coming. I hope you are studying. Make use of these lectures which we are ardently preparing for you guys, children. If you are not going to use it, then who will? So probably you should be studying right now. So keep on studying. Okay, now one more thing, very important thing. There is PU2 Success Blueprint Test Series, my dear children. Okay, and according to PU2 Success Blueprint Test Series, the test series have been launched on 15th of December already. Okay, so what you can do with this, you can still enroll, you will still get the test series and everything. We will be providing, we have already provided the solutions. Okay, there will be doubt clarification classes as well. So please do not worry about anything, we are there for you. And this course is for $599. If you're someone who is interested, the link is given in the description box, go for it. Yeah, and if you think that this video was good, do not forget to subscribe to our channel and show all the love, and wishing you all the best for your exams, wishing you all the love. Take care of yourselves, be happy, do not panic, everyone is there with you, okay, and you are going to rock, my champions. Bye bye.